And
Okay.
we will move to our next speaker.
Right.
Or Dr. Chan, yes, we have Cliburn Chan, and he'll be talking about the Center of Excellence for Multiscale Immune System Modeling, Modularity, and Bridging Across Biological Scales for Immune Digital Twins. Thank you for joining us.
Thank you. Can you all see my screen now?
Yes, I can see your screen.
Okay, great.
Looks good.
I'm really grateful to be able to give this talk and introduce the new Multiscale Immune Systems Modeling Center, which we call MISM. MISM. This was funded through a U54 mechanism. It's a new initiative from NIAID. There's only one award. It includes 3 research projects and 3 cores, and an opportunities fund for pilot work, which is a great way to collaborate with other people. It's just funded on 13th June, so we are 4 months old. This is a baby center. And it's quite exciting because the program office told us this is actually the first NIAID center that's been funded by all the 3 divisions as well as ONSET. We have a remit to operate across all of infectious disease and immune-mediated diseases. Broadly, I think MISM has two missions. The first mission is a research mission, so it's innovation, and what we are focused on is how to bridge across biological scales. We're very interested in how to bridge across immune biological scales. And this, in some sense, is ancillary to immune digital twins. But I think some of the technologies we develop could potentially be very helpful for immune digital twin development. The other more outward-looking mission that we have is coordination. And so, try to understand what all the multiscale modeling groups in NIAID are doing and see if we can coordinate the efforts better to build something more standards-based and more shareable and reusable. So in some sense, you're trying to break 2 silos. First, you're trying to break a vertical silo between investigators that just work on one scale, say molecular or tissue or individual epidemiology. And the second silo we're trying to break is we're trying to form bridges to multiple immune modeling networks. Organization. So the 3 cores, the administrative core, there's a community development educational CDEC core, and a model and data sharing or MDSC core. And then I'll talk a bit more about the research projects. So there are 3 research projects that try to bridge across 3 different scales. The first scale we are interested in is moving from individual antibodies to entire immune repertoire. The second one is looking at individual cells and molecules to tissue or organ scale. And the final one is looking at infectious disease transmission, trying to understand how individual immune heterogeneity contributes to transmission in a population. One thing to talk about the center is that it was designed from the ground up to be highly interdisciplinary. So in particular, every single core and research project is co-led by a computational scientist and an experimental scientist. And it brings people from a huge range of disciplines together, all the way from biostatistics to evolutionary anthropology. And it involves, I think, like 6 institutions: Duke, UNC, NC State, Penn State, Michigan, and UCLA. So very briefly, what does MISM have to do with immune digital twins? The thing about trying to model the immune system as an immune digital twin is that it faces the incredible complexity of the immune system. The immune system, as all of you know, is a complex adaptive system, which basically means that it's got many components interacting in nonlinear ways that result in emergent properties, especially across biological scales. So our job or responsibility is try to develop modeling frameworks that can bridge across these biological scales, and I'll go into more detail later. The fundamental premise of the sort of research that we're interested in is that high-resolution models are slow and expensive, but more accurate, while low-resolution models are fast and cheap, but less accurate. And so we want to learn how to combine them for the best of both worlds, right? And our approach is a learning approach. Can we learn emergent behaviors that matter at the next scale? And we adopt several different general learning frameworks to do this. The Project 1 uses a generative AI approach, Project 2 uses a scientific machine learning approach, and Project 3 builds a Bayesian framework to do this. So going into details in research project, the first research project it tries to bridge the scale from antibody to the immune repertoire. It's led by computer scientist Rohit Singh and immunologist Georgia Tomaras. And this is sort of like the exemplar for how generative AI might be used in immune modeling. At the granular high-resolution scale, we have atomistic modeling with molecular dynamics. So most of you know molecular dynamics are the people who tie up your entire computer cluster for 2 weeks to simulate 2 femtoseconds of what an antibody is doing. But one of the insights they have is that come out from small dynamics is that generally antibody binding to an antigen is not a simple process. There are many possible pathways to the bound state, as well as several potential intermediate, very short-lived states. And understanding this might give us some understanding of transitions that are necessary to go from independent antibody antigens to the bound complex. At the next scale, what we're interested in modeling the immune repertoire is basically using protein language models. I think most of you should know protein language models are basically large language models, except that instead of using word tokens, we use amino acid sequences as amino acids as the tokens, and try to learn basically evolutionary patterns conserved across proteins. In the case of antibodies, there is a non-evolutionary component, so additional tricks need to be implemented to make it work, but it can be done. And what Rohit has done is he's built an antibody language model, which he calls AB-MAP, which allows us to sort of like take an entire immune repertoire from BCR-seq or IG-seq and embed it in some vector space or embedding space. Which I would like to think corresponds loosely to the sort of shape space that mathematicians have talked about for decades now. And the bridging across skills idea is we know how to fine-tune, we know how to use granular information to make language models better, right? And one of the main approaches is fine-tuning. And so if we fine-tune for specific antigens from specific pathogens, we can get fine-grained local representation that we hope can improve the ability of the antibody language model to actually make predictions. And because it's a language model, it's obviously generative, so you can actually get the heavy chain and light chain sequences predicted by the model. Once you have these, you can engineer those antibodies and create them using recombinant protein technology, and then you can test them in the lab. And so there's a lab in a loop validation aspect to all of this. So what does this have to do with immune digital twins? I think quite a lot, actually, because I think the trouble with the immune repertoire is that it's insanely diverse and it varies so much from person to person. So in a recent study, for example, if you look at clonotypes of 9 individuals, only 2 out of 10,000 of the clonotypes that show overlap between people. So at the sequence level, there's very little overlap, which means it's almost impossible to train machine learning language models to predict what the repertoire will do. And if that's the case, then it's quite hard to imagine how you could actually build a reliable immune digital twin. But if you look at the embedding space of the immune repertoire, and the response to specific pathogens, they look very similar across individuals. So there's a huge collapse in the sort of structure function convergence. And I think this gives hope that with minimal one-shot or k-shot learning, we could actually use this sort of antibody language models to predict the immune response to a specific pathogen in an individual. And so I think this could contribute a lot to overcoming one of the complexity challenges of immune digital twins. I'll move on to the next research project, which is at a different scale. So here we are trying to move from a cell to a lymph node, and so understand this as a complex adaptive system with emergent properties as you move from molecules and cells to tissues and lymph nodes. And the sort of bridging technology one emphasized here is machine learning integrated mathematical modeling, nowadays known as scientific machine learning. It's led by mathematician Kevin Flores from NC State and Mika Luftig, a virologist from Duke. So one of the key aspects of being able to do this model is that we need attractable experimental systems. And nowadays, with the tissue on a chip system and organoids, we actually have really lovely experimental systems They are tractable in the sense that they are complex enough to have interesting emergent properties. At the same time, we can systematically perturb them, so we can actually evaluate hypotheses and check how our models are doing and iteratively improve our models that way. So this is an example of one of these non-animal models based on the emulate chip. And what the lab is trying to do is trying to grow a lymph node on a chip by taking lymph nodes either from people or from animals, dissociating them, and then reconstituting them within the chip, right? And so that the chip has inflow, outflow that you can monitor continuously. And when you run this system, what you find is that after seeding the immune cells, they spontaneously organize into follicular-like structures, which you can see in the lab chamber that you can track using live cell imaging. We are interested in actually a specific experiment involving reactivation of latent HIV. And so we also introduced engineered virus in this system, and antivirus is engineered so that it glows red when it first infects the cell, and then it glows green when it reactivates. So you can see at the individual spatial temporal level, individual cells lighting up and try to understand what's going on. Then we create high resolution models of this system using agent-based models. Here we rely a lot on the vivarium ecosystem and framework that Iran Ekman developed. The system goes really well with the Codex spatial proteomics technology that we're also using, because it allows us to identify where individual cells and molecular concentrations are. And so you can use that to sort of initiate and calibrate the agent-based model, which then together with expert knowledge and a lot of literature review to sort of understand the interaction rules, we can try to generate mechanistic hypotheses to understand what's actually happening in the in vitro system. The high level model are sort of viral dynamics models most of you are familiar with. They're very simple ODEs or PDEs. You can call it PDEs fairly easily. And then for bridging across scales, we are evaluating a few methods. So the standard way to sort of collapse agent-based model is using a mean field assumption, but mean field assumptions basically assume that there's no clustering, which is kind of nonsensical in this context. And so the mean field models don't work that well. And so we're adapting, we're trying to evaluate different scientific machine learning models to try to understand what's going on. And these seem to perform much better. So methods like SINDI or physics-informed neural networks. In terms of immune digital twins, I think this is probably the closest we get to actually thinking about how to build immune digital twins. And here we're not building a digital twin of a real person. We're building a digital twin of an in vitro system, in vitro model of a lymph node. So it's much less ambitious goals. And there are 4 steps to it. So the first step is, we have a tractable experimental system, which is in this case the lymph node on a chip. We can use that to build an agent-based model, and then we can generate lots and lots of synthetic data from the agent-based model. And that synthetic data allows us to sort of learn simplified ODE or PDE representations using equation learning, and then those predictions can then be tested on the lymph node on a chip, allowing us to iteratively improve our models. So in the long term, this system obviously allows you, to incorporate more and more live data streams, including live imaging, and you could do optimal control of the inflow of cells or media or soluble factors into the system. And so I think in the long run, this could potentially fit the full criteria for what an immune digital twin would be in a very limited setting. Research project 3 is moves from the individual to the population, and here we sort of want to investigate statistical methods, specifically a Bayesian framework for doing this. At the individual level, again, these are models you all are probably familiar with. We can model host virus dynamics, and sort of a little novelty is that because we have a very close relationship with the labs that measure immune functions, we can sort of put in parameters for things like neutralization, ADCC, ADCP, or CTL, and get the lab to measure them to some extent. And then what we're interested is like if you tune these parameters, if you tune them either singly or in combination, what is the effect on things like peak viral load, time to clearance of a viral infection? So this is a model that predicts basically viral load trajectory in an individual person. And then we position this person within a network, and so we want to understand who they talk to, who they interact with, and how these contexts are dynamic and made and broken over the course of infection. And then we use this sort of information to sort of build synthetic networks. Jim Moody is one of the people in our center who's helping us build this sort of synthetic networks and place agent-based models on top of them. These synthetic networks are based on very painstakingly collected real-world data. So, for example, here is data of the sexual transmission network through molecular phylogenetics studying the virus of people in Wake County in North Carolina. Bringing it together, we have we work with Jason Shi at UCLA, who's developed a really nice statistical model. It's based on the traditional SEIR model, so that's traditional ODE model, but it assumes homogeneity. And here we want to extend it in two ways, in several ways. So one is the first way is that this isn't the context unhomogeneous, but the context are on a dynamic network where the context can change over time. And the change could be driven, for example, by like what the model says about your interferon gamma levels that affect your sickness behavior and how you have to interact with people, whether you're symptomatic or asymptomatic. As well as the model could also tell you something about at this point in time, how infectious are you, or if you're the receiving end, how likely is it that you're going to be resistant to a new infection from a model? And so putting this in a Bayesian framework has the advantage that these models, we know how to calibrate them from data, including fairly sparse data. So it links back to possibility of capturing real-world data. In terms of the immune data twins perspective, I think this is a dream, but in the long run, I mean, every individual in this system could be modeled by a real immune digital twin, right, of an individual. And so there's a direct link to a real world, and you could populate the information with links to information from the electronic health records, for example. So that is the research. So the second part of the MISM goal is how to work with other research groups and research networks. And so I want to talk about opportunities we've been thinking of to work with MISM, or how MISM can work with other people as well. The model and data sharing core is quite heavily focused on learning about how we share and disseminate these models, and how we work with others to sort of develop the standards necessary to share. We don't want to reinvent the wheel, so we're working with people who've done a lot of work on this area already. The NIAID Discovery Portal, for example, the people in MIDAS. And then we'll be talking to Reina about biomodels and their experience in defining standards and sharing such models with people, as well as all the various standards that have been developed around agent-based models. And there we plan to work with people like James Glazier and Iran Ekman to sort of understand what it takes to actually build reusable agent-based models. The MDSC is also building computational platforms where people can run and evaluate the models, so that it's a common platform to support model sharing, and there are some funds to provide this service to the community. The Community Development and Education Core has also started work in the 4 months since it started. We've got a bunch of seminars coming up. Tal Aina is speaking at the end of this month on his models for influenza immunity across the lifespan. Jim Glazier is going to talk to us. He's visiting us and talking to us in November 11th on basically immune digital twins in the context of multiscale modeling. And then MIDAS will be talking to us in spring next year, but we haven't decided the date or the title yet. For summer 2026, we are opening internships for graduate students. So these are 8 to 10 week full-time summer internships for grad students in quantitative fields, so computer science, mathematics, statistics. And basically, the idea is that these graduates will be physically located here and will work with our multidisciplinary research teams on one of the 3 research projects. And so get to experience what team science in multiscale immunology looks like. And we'll give them probably co-mentors, one from biological community and one from experimental community. So again, they get a full picture of what's going on. And these are the potential research areas which are obviously closely related to our research projects. We are going to start opening pilot grants in November. So I think currently the idea is to open 3 pilot awards, and there'll be up to 60,000 each in direct cost for 1 year. And this is really very broad in the first year, all areas of computational immunology and infectious disease research. And basically, we want to know what other people out there are doing. It's interesting and fun in terms of multi-scale modeling, and we want to collaborate with them and work with them, learn from them, and share our computational resources and potentially our expertise in some of these areas with them as well. Coming soon, and all these announcements, I will post them to IMEC for circulation once it's up. Our website should also be up in November, but it's not up in time for this conference, unfortunately. And then lastly, we have 3 open postdoc positions. So anyone who's interested in any of these topics, is mostly mathematical modeling or mathematical modeling adjacent. We have 3 postdoc positions open. The selection committee will be meeting towards the end of this month. So do get your applications in early, and you can scan the QR codes if you're interested. And I think we'll probably be sharing the slides anyway, so you can look at them later after that. And yeah, we'd like to thank again IMEC organizers for the opportunity to introduce MISM, NIH, of course, for funding this. Actually, that's the wrong fund code. And the big MISM community, which you can see covers lots of areas and is growing. This is largely the community that helped put the grant together, but over the next 5 years, we expect and hope to grow the community to include many more people outside the research triangle. Thank you.
Thank you, Dr. Chan. Please put your questions in the Q&A. We have a few minutes. I guess I have a more general question. There are quite a few institutes and even some that may be popping out that want to create these platforms, want to be able to share. And it would be a shame that if everybody keeps creating new platforms or new ways to do this, how do we begin to sort of say what is already out there and how we can leverage what you've already done? So instead of I go recreate a brand new setup or so on, like how do we share with other communities what's already been done and make sure that it's not repeated?
Well, that's one of the things that MDSC is really trying to do, and through venues like this, right, conferences where we introduce MISM and sort of say that we want to learn what you already have, so we don't reinvent the wheel, and can we collaborate? We want to form partnerships. We would like to do as little work as possible because we're very lazy people, and we are very interested in how whether there are standards that we can adopt that are suitable, and so we don't need to do new things. I think outreach, once our website is open as well, we will be advertising that we want to collaborate with people to build standards and share platforms and so on. The platform that MDSC is building is a fairly general platform. You can put any models in there. It's not really meant to compete with something like biomodels, but it's more like a place for us to showcase, like the pilot grant awardees as well as our own research projects, what they've done, and so the MISM will support them to make that public and accessible to everyone.
Thank you. There's actually 2 questions. One of them says, how does the epi modeling differ from the epi-sim? That's by Amber Smith.
Well, I'm not an epidemiologist, so I'm not really sure what the EpiSim project is about. I think our focus is on how do we bridge across scales to understand the content of individual heterogeneity. Again, that may be quite closely related with what EpiSim is doing. I know there are other groups that are doing such things. In fact, later this month, we're meeting Justin Lessler from the Insight Network, who's also building a lot of immunological epidemiological simulations. So again, we'll see what we're doing, but we're very focused on the social network aspect of it because Jim Moody and Charlie are sort of experts in that area, as well as on the Bayesian aspects of it. So, and Amber, we can talk offline as well, and you can teach me much more about EpiSim.
Thank you. There's another question that we'll probably need you to, it says, for postdoc position, what is a background requirement? I'm assuming you're going to have to send them additional information. So if you don't mind answering that on the Q&A.
Sure. And the QR codes will bring you to the actual list of requirements and expectations for the position. So I can share my slides with you, and maybe you can share and people can access it later to get the QR codes if they didn't get a picture there.
Yeah, I believe this is recorded and will be posted
Okay.
at some point in the future. So
yeah, and if you cannot get access to them, just email me directly.
Thank you. Dr. Chan, you have another question now? It says Alexander Silalahi says, thanks for the talk. How much do you see a practical identifiability problem as you try to bring the ODE models into clinical data?
Yeah, we see huge identifiability models. There's a lot of work going on in sensory analysis and uncertainty quantification within that particular modeling group as well to try to approach this problem.